Hi guys, so in this video we are going to talk about the difference between the cellular immunity and the humoral immunity. So we're going to start first with the cellular immunity. So let's define it. The definition of cellular immunity is an approach mediated by T cells which recognize the presence of the antigen. They bind to the antigen and trigger a response by the other immune cells. T cells get their name from the place where they mature. The T cells mature in the thymus area of the body. T cells are produced in the bone marrow but then enter the bloodstream and travel to the thymus for maturation or specialization. There are two major types of T cells that destroy the antigen. So, number one is the regulator cells. It's going to be the helper T cells and the suppressor T cells. The second type of cell will be the effector cells or killer cells. The T helper or TH cells contain a protein called CD4 plus, which is why they're called CD4 plus cells. And with that, there's four types of TH cells that have been identified. So first one is the TH1 cells. It participates in inflammation by producing cytokines as well as stimulating macrophages. The second one is TH2 cells. This one activates and call up the B cells to produce antibodies. The third one is the TFH, or it's also called T follicular helper cells, and it assists the B cells as they develop into antibodies secreting plasma cells that is located in our lymph nodes. And then the last one will be the TH17, which is involved in inflammation and protect our surfaces of our bodies, such as our skin, the intestinal lining, against bacteria. The suppressor T cells turn antibody production off. The second type of cells is going to be the killer T cells. It's also called cytotoxic cells. Its aim is to destroy cells infected with viruses by releasing lymphokines that degrade cell walls. It also have a CD8 plus protein on their surface, so they are often referred to as CD8 plus cells. T cells work to protect the body against intracellular viruses, fungi, parasites, bacteria, and cancer cells. They are also responsible for hypersensitivity, which is a different topic, and a transplant rejection. T cells can detect intracellular pathogens because infected cells can display peptide fragments derived from the pathogen's proteins. These foreign peptides are carried to the cell surface by specialized host cell glycoproteins, referred to as the MHC or major histocompatibility complex molecules. T cells can recognize these small peptide fragments bound to the MHC molecule as an antigen. So now we're going to talk about humoral immunity. So B cells mature in the bone marrow and they differentiate into either memory cells or Ig secreting antibody cells. These cells eliminate extracellular bacteria, neutralize bacterial toxins, prevent viral infection, and produce immediate inflammatory response. Each B cell has receptor sites for a specific antigen. Where it encounters this antigen, then the B cells gets activated and multiplies into either antibody producing cells or memory cells. Other info to note is that these cells has a 24-hour lifespan, but they can begin the antigen production within 72 hours after the initial antigen exposure. So the next time somebody's exposed to the same antigen, the response is going to be a quicker action. Immunoglobulins. So there's 5 Igs. So we have IgG, IgM, IgA, IgE, and IgD. So we're going to go over first IgG. So it's the main defense against bacteria. What's special about this, because it allows the Ig to cross the placenta to protect the fetus against infection. The next one is IgA. This one is found in the membranes of the respiratory and GI tracts, and then tears, saliva, colostrum. It basically protects the body from external exposure. The next one is IgM, which fights the blood infections and triggers additional production of IgG. It is present in lymphocyte cells, the first antibody made by a developing fetus. The fourth one is IgE. It is the one that protects the body through its presence in mucous membranes and skin. It also triggers allergic reactions and responds to parasites. Again, it also works hand in hand with the eosinophils. And the last one is going to be the IgD. This one is present in blood serum in small amounts and on B cell surfaces. It is also a receptor for antigens because it helps anchor cell membranes.